So I want to thank the earlier speakers for doing all the heavy lifting. They covered all the technical stuff. So they left me the more fun and lightweight stuff. So I'm going to be covering quite a number of items. Hopefully you will not fall asleep and hopefully it won't be too long. So biological pathways, network, and towards the end, gene ontology. Most of this you should be familiar, but hopefully you still learn something new. Okay, so we get started. Now, if you're wondering in the realm of central dogma, where does this all fit? So it fits somewhere there. Once you have done all your assembly, variant mapping and all that, and you have your genes and proteins, gene annotation, all that done, then you can do the downstream stuff. So I consider this as part of the downstream analysis. So you have your assembly and mapping, then you come to the fun part. So biological networks, biochemical pathways, we look at them here to better understand live activities. So I'd like to begin with the definition of a network. Now, this is a general definition. It's a little mouthful, but I've highlighted the key aspects. An organized deterministic system, so there's some sort of an organization. It has many structural components, can be units and subunits, working in a concerted way, in a certain programmed way to perform certain functions. And there's some rules that is being followed. So that's a general definition of a network. As for biological network, but actually before I go to that, usually when you think of a network, you see a spider web or you think of a mesh with a lot of connections. So it's something like that. So biological network is basically the same thing, but applied to life activities. Okay, so we'll see more about this later. Now, biological pathways actually are a type of a network, but they are directed. So there's a direction involved. So biological network is a much larger, you could say, phenomena or concept, and biological pathways are a little more focused in the sense that it's directed. So as part of the definition, a series of actions. So you see, a certain number of steps that need to be followed, which happens one after another. Now, there's some basic terminology or concepts when you deal with network. There's this term node. And this could be basically something that symbolizes your gene or your protein or whatever is the item that you're studying. And it's a one-dimensional representation. And your pathway is just a series of interconnected nodes. So this can be two-dimensional. A network then is something more complex, interconnected pathways involving, let's say, various interconnected pathways, the nodes of those pathways coming together, some sort of an interactomics, and this is multidimensional. And the connection between them is called edges, and these are based on certain evidence, and it can contain weights or even direction. Now, just to compare the two, so gene networks, these are cluster of genes, as the name implies, with some evidence of co-expression or whatever the evidence may be that is the basis for that connection of the edges. And so the connections represent the degree of co-expression. In this case, if your evidence is co-expression, and in-depth knowledge of the process is not really necessary, and they are largely not predictive or of lower predictive power. On the other hand, pathways is a series of chemical reactions or steps. The correct connections represent the relations between the molecules, so there is an actual relationship. A lot of this is involved in you see a lot of this in metabolic pathways, so enzymes are regular components of those pathways, and they're highly predictable. So the predictive power is rather high in contrast to this. All right, here's an example of a gene network, so with low predictive power. And of course, you still learn a lot of things. You still see, what nodes might be playing a more important role. If you have a node with a high degree of connection, if you were to delete that, or if there is a mutation in that particular gene, then a lot of different nodes would be impacted and affected, right? So there's a lot of value coming from this. And here's your typical pathway. This is glycolysis, so which we all are very familiar with. Okay, and of course here, if you were to perturb something at the top there, then you know what's going to happen. So it's going to really affect the downstream. In a nutshell, then, if you were to compare these, in terms of curation, gene networks are relatively easy. They can be automated and can also be manual. These are much harder, and they are mostly manual. The nodes, in this case, would be the genes, and here can be any general molecule. The edges, which is the evidence for that connection, can be co-expression or quite a number of different items. But here's going to be mostly, quantifiable connections, evidence of connection between the items, the entities. Low fidelity, high fidelity, low predictive power, high predictive power. Here's an example of a network, another example, and this is just, in the larger scheme of things, a pathway among many other pathways. So now that was just a quick overview. I'm just going to dive in a little bit into biological pathways and then biological networks. So there are many examples of biological pathways. Metabolism is one of the popular one. You also have genetic information processing pathways, environmental information processing, cellular processes, organismal systems, human diseases, drug development, etc. Sorry. Right, so if you were to go to the KEGG database, you actually can get a very comprehensive list of different pathways, biological pathways that are available. As you can see, there's just too many of them there, right? So, and this has been organized, curated, and you can actually click on each one of these and really see the pathway. Very valuable resource. And there are many repositories. KEGG is just one of them. There are a few others, in case you're not aware. So you have BioSeq, MetaSeq. So MetaSeq is usually mostly, I think, for metabolism. BioSeq is much larger, and I just read earlier that they are now also handling microbes. So this also has pathways of microbes. KEGG is the rather popular one, but I think it's mostly reference pathways. You have BioCarta, where you see those cartoonish pictures, and biomodels are mostly mathematical models. Here's a comparison of the four major databases. So in terms of curation, this is manual and automated. KEGG is rather automated, manual. These numbers are a little outdated. I think in terms of, so this is quite broad number of species. Even I think the latest number is 20,000, especially when they, now that they're adding microbes. What else? So this one is mathematical. It's mostly model. This one, I think, it is quite broad. Can't remember the specifics of what they are interested in, but these are the few major ones that you can explore. I mostly use KEGG, but you should also explore. In fact, a lot of the tools that we use, they do actually take advantage of the multiple databases, which is nice. So you don't have to yourself kind of figure out which one to use. So KEGG, just to give you an idea of what it looks like in case you have not been to. So this is an old picture of KEGG, and you could click on the pathway there and it'll bring you to something like this. And this is, although it looks static, you could actually click on so this is the one that you searched for, it's in red. And if you were to click this, it'll bring you to a record where you get to see the list of other pathways that this particular gene is involved in, and then you can jump to the other pathways. So you can keep jumping from pathway to pathway and learn quite a number of things. And these lines, they do have meanings, and if you click on the help page, you get to see that they represent activation, repression, inhibition, etc. Right? So it's not really a picture from a book, but it's something that is very informative in terms of what's really going on. All right, so why do we do pathway analysis? Well, for a number of reasons. It's the logical next step when you have data from high throughput experiment. You want to make sense of what's going on, and pathway analysis can help you understand. Because at the end of the day, these molecules, they don't work in silo. They are connected in a certain way, and and bring out an effect, which results in activities. So you want to see that relationship between these different connections. So the high throughput, they don't really produce biological findings. They don't work alone. And so they actually help you interpret and give you a global perspective of what's going on. So this is a very nice review paper by Atul Bute, and this was like this in 2012, and they reviewed the last 10 years since then, from then, so 10 years before that. And what they have shown is that, the pathway analysis has kind of evolved over 3 generations. Started early on as typically being over-representation analysis. So if you remember, or if you heard of people talking about fold change, 3-fold change, 2-fold change, and you identify differentially expressed genes. So that's what they were doing. Something was overrepresented. And this analysis typically involved your DEGs. So you mostly focus on your differentially expressed genes, and you kind of discard everything else, right? So whatever you thought was interesting, and that's what you take forward. And the FCS, which is like the 2nd generation functional class scoring, this one, the main difference between this one and the earlier one is that instead of just focusing on the DEGs, the differentially expressed genes, you actually take everything, right? So you start analyzing everything because, there are certain, the other genes can't be just playing no role. They must have some role, maybe, a little less significant, but everything kind of adds on. So the add-on effect of all that was important and needed to be studied. So this allowed you to overcome that limitation and take everything and then study that and find out what we can learn from that. So the 3rd generation basically recognized the limitation of this, which is that these genes, they are not just connections, they actually have directions. They, whether they're upregulating or down-regulating, and they have certain number of reactions, position of the gene, the types of the reaction. So the pathway topology, type of tools, what they do is they consider these additional factors, additional aspects of the data, and incorporate that to better understand what's really going on, right? So you can't, for example, here you say, okay, these are upregulated, downregulated, but are they upregulated, downregulated in the same direction of the pathway, or they could be opposite direction of the pathway? And by knowing the directions, we can maybe learn something else, right? So that's the 3rd generation. Maybe there's a 4th generation. I've not seen an update to this, or maybe there is out there. But this gives you a nice overview of how the analysis has evolved. And typically, your input is your high throughput data. In this case, could be your expression data, and you also kind of club it together with pathway database, where the annotations are brought in from the pathway database. And you pull all that together, and at the end of that, you learn about the significance of a particular pathway. Some other terms that you may have heard, pathway enrichment. So which pathways are enriched by these genes that are playing an important role, and then you focus on those pathways to try to understand what can you learn from those pathways about the biology of what's going on. It's quite powerful. I remember a colleague from Hopkins where he was studying sleeping sickness. So, you know that sleeping sickness involves this trypanosomes and involves the blood-brain barrier. And if you didn't know any of that, your pathway analysis actually directed you or pointed you towards pathways that are involved in the brain. So if you had no idea, the data kind of tells you something. So there's that strong correlation, and it's quite significant in terms of what you can glean over from the data. Okay, so this is just an overview comparison of the 3 generations of approaches. So the pros of ORA, simple and powerful, requires less input data, less because you are focusing only on the DEGs. Con background, you make a lot of assumption, you discard a big chunk of your data, and you assume all genes are independent. So you ignore interaction between the genes, and you only assess the number of significant genes, and you end up with many false positives. With the FCS, where you take everything, is definitely more accurate, and it uses the entire list. It still does ignore the interaction among the genes. It analyzes the pathways independently, and you still have many false positives. So the 3rd one, the topology of the perturbation type, you consider each gene's specific role, which I mentioned earlier, position, magnitude, interaction. It is of higher predictive value. It requires more data and takes slightly longer, and At that time, it doesn't seem to be, was not seem to be applicable to metabolic. I'm not sure at the moment whether it's still not applicable. Here's actually a list of tools for these 3 generations. So ORA tools, FCS tools, and PT-based tools. So it's a very nice review in the sense, they provide you a list of all these different tools that you can explore. I want to highlight two of these tools. David, many of you may have heard or have used. So I think David is mostly the 1st generation ORA, and it also provides ontology information, which I'm going to talk about later. So this is one example, and this is of course freely available. I want to contrast this with IPA. Many of you may have heard of IPA, Ingenuity Pathway. It's actually very powerful, but it's commercial. There's just so much information, especially on the genes in terms of perturbation. So you can use this to do perturbation. Luckily, they give you 2 weeks of trial. So if you have multiple email accounts, you can just try. And I know some people do that. They publish with that analysis they do. And what's interesting is if you Google for David, you will get an ad from IPA. They're so aggressive. Replace David pathway, try IPA. All right. And if you're searching for KEG, they will say, replace KEG, use IPA. So I guess they have too much money to pay Google. All right. So so that's a bit on pathway analysis. Moving on to biological networks. So there's so many different types of networks. You have intracellular, and under that you get protein interaction, metabolic, et cetera, intracellular, then intercellular, organ, tissue, ecological, and evolution. All right. For example, if you were to go to the protein interaction networks, where is the data coming from for that connection, for that evidence, for the edges, right? The lines that connect them. So those evidences come from your wet lab experiments. So wet labs are very important. Of course, it can also be predictive. Here's just an example of some of the wet lab where that data comes from: yeast-2 hybrid assay, syn expression, mass spec, etc. You also have in silico prediction as part of that. So how does one analyze the network? You use something called graph theory. You may have heard of this. This is not something new. Actually, it goes back to something very old. So the foundation for this predominantly seems to be laid down in like 1700, maybe earlier than that, but this is the most notable one. So this person, Leonhard Euler, from Königsberg, it's part of Russia now, previously Prussia, and basically he had this question, whether it's possible to walk with the route that crosses each bridge exactly one. And what I've heard, what I've read is that he was disappointed he couldn't do that. And some people say that the problem was that he didn't have the tools to analyze and figure out what would be the best way to do that. I'm not sure whether it's still possible, but we do have the tools, and I need to maybe find out whether somebody actually did it. Or you could tell me if somebody has done it. Now, there are a few types of network. You can have undirected ones, and you use something called adjacency matrix to kind of map that picture into a matrix that maybe makes sense, or that describes what's really going on. And it's rather simple. So you take V1, so you put all the components here, all the components there. So V1 interacts with V2, interacts with V4. So V1 is 0 with V1 because it's itself. It's not interacting with itself. With V2, you get 1. With V3, it's 0 because there's no connection to V3. With V4, it's 1, yes. V5 is 0, so it's just that, right? Now, if you go with the directed one, so something interesting is happening. Now, V1 is interacting with itself. So V1 to V1, it's going to be 1. V1 is interacting with V2, so you get 1. But no V3, no V5, and with V4, so you get 1. Now, where is the direction? The direction is this. So now if you take V5, so if you go down to V5, V5 doesn't interact with anybody, from its point of view. And it only interacts with V2 from its perspective. So V2 will have 1 and everybody else will be 0. Okay, so that's the directed one. Weighted one, and of course you could have weighted, undirected, weighted, directed. This is undirected. Weighted, instead of putting 1, you simply put the weight that you have assigned for the edges. All right, so in this case, V1 to V2, instead of putting 1 there, you put 5. Okay. All right. So now I just want to maybe just share with some properties or characteristics of these networks. So there's this concept called small world, which, sometimes it's an expression we use a lot. What a small world, when you meet people who know somebody you know. All right. So small world is a type of a mathematical graph in which most nodes are not neighbors of one another, but the neighbors of any given node are likely to be the neighbors of each other. I think you may have also heard of 6 degrees of separation, whereby we are kind of connected with everybody through six connections of our own, right? So that would kind of capture everybody else out there. And to achieve this, there are 2 ways to achieve this. One, opposite ways of achieving small world. The maximum linking expands, basically you kind of link everybody to everybody, and this is called a click. Or the other approach is, you maximize individual interaction where one is just, connected to as many. So that's the star graph. And then these are some of the common topologies that you might see. You have ring, mesh, star, fully connected, that's a click. It can be a line, can be a tree, can even be a bus. All right. And I just want to use this just as a fun activity to kind of highlight something. Let's say we had SARS corona, well, we are still in the pandemic, and let's say this represents a network of the virus, let's say a protein of the virus, and what it interacts with to result in infection. Now, if you wanted to perturb this network, if you wanted to disrupt this network, what would be your point of attack? For which node would you develop a drug? Any thoughts? I know it's kind of hard to point, but maybe you can just shout out and describe. Which one would you? Okay, well, let's do one by one. Okay, what about this one? Will you perturb this one? No. Okay, why? Because it's only going to impact this one, and everybody else is still going to be together. What about that one? No. What about that one? I guess you know the answer. That one, right? It's just going to cut it into half, and maybe it will affect the survival of the virus, right? So this is one of the ways you can utilize information from here to understand the biology. All right. Now, there are a few other terminologies like size, the number of nodes, density, the number of ties that are present versus blah, blah, blah. Degree is a very common one, basically the sum of connections from an actor to others. How many total connections do you have? And you also have something called in degree, out degree. So this is basically in degrees coming to you, out degrees from you to the other actors. Diameter is the maximum greatest, you can read that. And walk is basically the path you take to go from one point to another point. Geodesic, I'm not sure if I'm pronouncing it correct, geodesic, okay. Distance is the shortest path that you would take to get to a particular point. Maximum flow, the number of actors you would have to go through to reach a particular point. And these are some other terminologies: degree centrality, closeness centrality, information centrality, betweenness centrality. What we did earlier is more like information centrality, all right? So which one, a node which plays an important role in maintaining the robustness of the graphs. And if you were to attack that, then your network would kind of collapse, all right? So this is also something you can use. These are terminologies that help us communicate with others when we are dealing with these kind of graphs. This is a very interesting one. We talk about essential genes and non-essential genes, and if you were to plot a network of those genes, then you see something like this. In terms of degree, now degree is the number of connections. So you see essential genes, they can have up to like 300 different connections, but very small number of them. And you have, of course, few of them that have very high frequency, whereas the non-essential genes, there is some overlap, but after that they taper off, so it's up to around 150. Actually, one could actually use this to determine whether a gene is essential. So if you have something with, let's say, more than 150 degree, then it could be an essential gene. Of course, below that it's hard to say because there's a significant overlap. Right. So these are some of the some aspects of the proteins or the genes that we are studying. We can learn about them by using tools like this. Now, there's also this concept that, it's not the overall network topology that's only important, but also, the subcomponents of that. So often you have something called modules or motifs, submotifs, and they can be rather, smaller in number of permutations that you can have. They can be rather like your Lego blocks, you only have like few combinations. So they can also be very useful in helping us understand like motifs and domains of proteins, understand what's going on. In this case, you have like 209 of bifan motifs found in E. coli regulatory network. So also do consider focusing on the modules and motifs when you are analyzing this. Now, Cytoscape is a very popular software, freely available for you to do this kind of analysis. And it's freely available, open source, etc. Very visual, helps analyze. What's the best part about Cytoscape is it has some sort of like its own app store, right? So you can kind of install plugins to do different kinds of analysis. So these are some of the different tools that you can bring in, add on, and use them to analyze. So you have MCode, which is very popular. You have Bingo, etc. And Gene Mania, I just wanted to use this to highlight something, that Gene Mania, which is also found in Cytoscape, you can use it within Cytoscape, but there's also a website, Gene Mania. You could in fact go to it right now. Maybe let's just try, since all of you are here, just for fun. Go to Gene Mania and just type P53 and see what happens. So hopefully it won't take long. And I just wanted to highlight how much work has gone how much work has gone into making something like this possible. And this is taking advantage of the information available in the literature. And you will be amazed when you actually click on these evidences. So you can see from here, for P53, most of the evidence is physical interaction. And that's 77% of the evidence. You do have some other evidence, but this is the large majority. And if you were to click on that arrow there, you'll see all the list of papers that have actually contributed to that information. That's a lot of work in terms of curating, finding those papers, and then making use of them to be able to plot something like this. Just a fingertip away. So, and this could have been like the PhD of maybe 5-6 students in the past, and now it's just available to you, just a click away. So do take advantage of this, but this is also available in Cytoscape, and you can do so much more with Cytoscape. Moving on to the last part, gene ontology. This is, I think most of you are very familiar with the gene ontology, the 3 components, so we'll come to that. But I wanted to cover some aspects of the concept of ontology. So this is what I'm going to talk about. This is just a quick outline, standard nomenclature, the limited computational value of free text, and CVs and ontologies. This is control vocabulary. All right, so standard nomenclature, you probably can relate to this, a plague of biology. Names are just randomly given, and you have all kinds of funny names. Sometimes, names of proteins are based on the tube, the labels of tubes from which, they were isolated. So this is just to demonstrate that. So P53 has like 3 different names. This one has that many names. This one has that many. And you can also see that P38 is being repeated. And if you were to do a PubMed search with P38, which gene are you really going to get? And are you really studying the right gene? So you got to be really careful here. And luckily, this is not a problem that we are just realizing. There are already various groups. I think Hugo has been looking into this and has been trying to address this issue by coming up with official names. So how do you overcome this? So when you are publishing papers, try, besides using a common name, when the first time you declare that particular gene, put the accession number somewhere so we know what are you really talking about. All right, so use identifiers, database identifiers, when referring to a particular database entry. And of course, please include the official gene name when you're describing your research. And you can go to Hugo Gene Nomenclature or Hugo database to kind of get the official gene name. Or you go to the gene database at NCBI and you'll get the official name. Now moving on to the next item, I think we all know this, that free text has very limited computational value, and you can see where this is driving to. So text mining versus human interpretation of free text, of course human will win. But there's a lot of value that can come out of this if we can standardize come up with a certain structure for our free text. So for example, we would have something like this, which would be easier for a computer to assess. Molecule A was found in the cytoplasm, whereas molecule B was not. Rather, it continued to accumulate in the nucleus. On the other hand, we have something very simple: molecule A cell place, molecule B cell place. So something like this would be much easier for you to kind of navigate computationally, extract information, parse the data, do so much more gymnastics with it than something like that. All right, so we need to come up with control vocabulary. This is particularly important when we have a lot of synonyms for something that we are referring to, and we also need to define come up with ontology for biological data. So what is control vocabulary? A set of a set of official descriptors assigned to a particular entry. I want to focus on this, illustrating the relationship between synonyms and preferred usage terms. So control vocabulary is meant to handle those nomenclature issues. Ontology, on the other hand, is conceptualization of knowledge of a domain. You are basically putting is a control vocabulary that describes the objects and the relationship between them. It's a mouthful. So these terms are sometimes used interchangeably. Think of ontologies as hierarchical CVs, control vocabularies that specify a relationship. I'm going to use a grocery shopping database as an example. So we may use, for example, different words for the same thing. So bread is in English. I don't know, this is what French or German, Le Pen, das Brot. So they mean the same thing in different languages. And ontology would be to formally classify the concept of bread or the relationship between different aspects of breads. So here's just an example. If we were to come up with this classification, okay, a bread is a grain product, and it can be broken down into breakfast cereal. And on the other hand, you have bread, and these are the synonyms of the bread. And bread can be further broken down to loaf bread or flat bread. And flat bread, you can have roti prata, pita, naan. Roti prata, you have to go to Malaysia to try that. All right, but you can, if you go to the Indian shop, you might get that too. And then loaf bread is white bread, synonym to Wonder Bread. So now that we have a classification like this, it becomes very powerful. If somebody who didn't know these concepts and they, let's say they started off with naan and they had access to something like this, they would immediately know that naan is a flat bread, it's bread in general, and it's a type of a grain product. Right? So much information that we can gain through something like this. So the same idea was to have something like this for genes. So we have the gene ontology. So it's a database of terms to describe gene or gene product information, the terms that are applied to gene products. So we can have a common language to describe these biological observations, and we can do some computation on those. So you know the three components, so cellular component, biological process, and molecular function. So you can, basically we are focusing on three aspects of genes. Where are they located? What do they do? And what is the process that facilitates that mechanism of function? And here's an example of an ontology, gene ontology. So it's part of cellular component, and you have these different components, and you keep going down. You define all the different components. At the bottom, you'll have your genes being listed. Then you can go backwards, and you can go to the parent term, or you can go downwards to the child term. This is more granular. This is less granular, and these can actually help you learn a lot about your gene. So if you found your gene to be here, then you know that it's an organelle, and then it's located here, it's located there. So here's an example for P53. If you go to a particular gene or protein database, you get to see the list of these gene ontologies, especially the highly enriched ones like RefSeq or UniProt. They are very good at populating the data, the record with all of these. So you get to see all the different lists of these terms, and you get to learn about what your gene might be involved in. And in general, the more specific, more granular term that's possible is applied given the evidence. Now here's a web server that you could use called Amigo to kind of check out a particular geo term and see what is it all about. If you had a particular geo term you wanted to know more, you could come here, and it will also provide you with the actual breakdown. All right, so now I want to showcase some applications of some of this. So for example, we could actually, one way is to go down and be more specific, where you could also be less granular, and there's some application of that. So for example, protein A is found in nucleus, protein B is found in nuclear matrix, protein C is found in mitochondria, and you wanted to be able to describe, come up with a term that would describe all the three maybe at the same level. So can group by a common parent term. So, and that common parent term turns out to be intracellular membrane-bound organelle. So you can say that protein ABC are intracellular membrane-bound organelle proteins. So, it facilitates, it enables you to be able to use these ontologies to describe the proteins that you're studying. And it also tells you something about the role of that particular protein, the proteins that you're studying. And here's another application. Let's say you go to PDB, and many of you do MD and docking and all that. You use PDB a lot. And let's say you had this complex query that you wanted to do, which is going to be difficult to do using keyword search. Let's say your query was, show me all of the structures where one or more of the molecules can reside in an intracellular membrane-bound organelle. As I said, that's the kind of question you have. Now, PDB provides a breakdown of its records according to these ontologies. So you would go to that particular ontology term, intracellular membrane-bound organelle. You click on that, you'll get all the records that are local, are involved at that particular cellular component. So it's very powerful in that sense. It really enables search that's beyond basic search or advanced search. That even advanced search would not be able to allow you to do something like that. And I'm not sure whether you realize, actually, a similar concept is being used by taxonomy. So taxonomy is using a very similar concept, and almost every record in NCBI has this lineage, the taxonomy lineage. And using that, you could actually, for example, come to the taxonomy browser and type human, and you get the lineage. And let's say you wanted the data of all mammals. Then you just need to click on that, and it'll give you a breakdown of all the data for mammal. So you don't even need to do a keyword search. And when you do keyword search, false positive problems and all kinds of other problems. So this is very powerful. And then when you just click on that, you'll be able to get all the protein records that have mammalia listed as part of the lineage. And of course, you could say, okay, I wanted all records involving cellular organism, or all records of eukaryotes. You just click on that, and it will change, update, and then you click on the number, it'll bring you to the respective database, and you have it. You can do a lot of analysis using that data. All right, so grouping entries, here's just another example of an application. Find me all of the protein interactions where one protein is from a virus and the other protein is from a mammal. Now that doesn't seem like a simple query, but you could come to, let's say, bind database. You say find all records. On one hand, you have the taxonomy lineage virus, the other one mammalia, and then you'll get the hits that are, going to have that kind of interaction. All right, so that's simple application of Go, the concept of Go, but you can also have some sophisticated applications like analyzing expression data, evaluating protein-protein interactions, especially when you use David or some of the tools I mentioned earlier. They also bring in these ontology terms to help you better learn and know about your genes. Since, well, in those experiments, you'd be studying your genes anyway, and you have gene ontology to take advantage of. And here's just some other tools. For example, MapFinder, a tool for mapping gene expression data to geo hierarchies. You also have something that was mentioned earlier by, I think, Dr. Nick or Jeremy, Blast2Go. So you could also take advantage of this. Basically BLAST, but for Go terms. All right, so now Go terms, some of them are more common than others. So it could be, there's this chance of being a randomness. So the more common Go terms will occur by chance. The question is the term more common in a gene list than expected? So is it really significant, or is it just there because of it being more common? So you can actually ask such question and use, for example, Cytoscape's Bingo plugin to ask that question and find out whether it's really significant or just by chance. All right, so I hope I was able to give you a quick overview of GEO. And in general, then, the value of CVs and ontologies, it increases our ability to retrieve records, and it also groups observation at multiple levels, so we can learn as much as possible about all these if you're studying genes, then genes and the relevant terms available. All right, so that's it. That's all I have, and I hope this was useful. So just a quick recap. We learned about network and pathway in general. Then we also dived in. We explored a little further about the different databases for pathway, the different types of networks, and some features of these networks, some terminologies, and lastly, the value of CV and ontologies. Thank you very much. Questions? Yes. Yes, you can. Question was, she has never used Cytoscape before. She has gone to other databases like Spring, protein interactions and all that. Question is, can I use those data, plug them into Cytoscape and make a network out of that and see what's going on? Short answer is yes, you can do that. Okay? Any other question? Yes, Amit. You mean? Yes. Okay. Well, as far as I know, I think these are already well defined, predefined by the database. So if you type something else, it will probably not work. So you have to use that. I think, but your question is whether it's just like PDB has 4. Yeah, so I think they have maybe a fixed size of that accession number. It might evolve if they discover something, but I think it's pretty well defined. I think all these three, function, biological process, cellular component, I think they've kind of mapped everything that we might need to know. So I don't think there'll be a sudden tsunami of new components, new functions, or new processes that they would need to expand. And I think within whatever digit they have, there's still scope for that to be expanded to accommodate any new ones that might come along. Any other questions? As everybody says, we can always chat later. So thank you very much.